Hey guys, so we're going to talk about chapter 6. So this chapter goes over bacterial growth, their nutrition, and how they differentiate. So we're going to go over starting with their nutrition. Oopsies, if I can get the computer to work. There we go. So, microbial nutrition. So, are there any limits to bacterial growth? And the simple answer is yes. So, even though bacteria are pretty simple as compared to eukaryotic cells, they are very efficient in replicating themselves, such as E. coli. So, we saw E. coli today in lab. He grows extremely quick. So, after like 24 hours of incubation, you can have millions, billions, maybe even trillions amount of E. coli cells in your culture. But bacteria, it is limited by nutrition. So you can, oh, excuse me, you can limit bacterial growth by limiting how much food do you give them, as well as their environment, such as temperature or their acidity. Also, some salt. So, some bacteria don't like extreme salt environments, and we'll actually do that later on in lab. I believe it's experiment 5. We'll go over that. But just know that yes, you can limit bacterial growth in numerous different ways, and nutrition is one of the main ways. So, essential nutrients. So, these are the compounds that microbes cannot make themselves, and they have to gather from their environment. And these are just some of the basics. Also, there are some traces of like additional cobalt, magnesium, nickel, and zinc. But the big players, though, guys, is carbon, nitrogen, phosphorus, hydrogen, and oxygen. Those are the biggest players that they can gather very easily from their host, such as us. So just know that there are essential nutrients that a microbe has to have, just like us. And what makes them an essential nutrient is the fact that the bacteria cannot make it themselves. So many microbes, they can't assemble these compounds that they need in order to grow on their own. So what they do is that they have additional growth factors from the environment that can help them out, such as the influenza. So that's the causative agent of meningitis, and it can't synthesize NAD or the heme, so a V factor or an X factor. So with that, it cannot grow in a laboratory setting. So with that, what you have to do is that you actually have to add that into the media in order for it to grow within culture. So meaning, so as we saw today, like we had some nutrient agar plates. So those are plates that pretty much grow anything and everything because it's like the perfect little environment and food source for the bacteria. But certain bacteria have to have something added to that nutrient plate in order for it to grow. And this is an example. So what you can do is you can add or take away stuff so you can affect microbial growth. And we'll actually play with that a little bit in lab later on in term. So microbes, if they are obtaining carbon from an outside source, they're called autotrophs or heterotrophs. Oh, excuse me, guys. So heterotrophs, they specifically rely on other organisms to make organic compounds for them to use. So such as us, like we are heterotrophs. Like we have to get our carbon source from a lot of times sugars, like carbohydrates. So we are considered heterotrophs, and there are quite a few bacteria, just prokaryotes in general, that are considered heterotrophic. And then there's also autotrophs, where they actually use the carbon dioxide and they are discarded by heterotrophs to make cell constituents that are made up of carbon, hydrogen, and oxygen, such as carbohydrates. So autotrophs, an example of those can be, I'm blanking. I'm sorry, guys. I want to say a plant, but it's not a plant because that's a phototroph. I'm sorry, guys. I'll come back to an example. I'll probably come up with one in class. But autotrophs, they make their own food essentially. So autotrophs, they make their own food, and then heterotrophs eat the autotrophs or eat the phototrophs. So phototrophs, like I said earlier, those are the ones that gain their energy from absorbing light from the sun. So that's an example of like plants. Plants do that a lot, or any photosynthetic organism. So some bacteria are considered phototrophic. They're not a lot, but there are a few. And then the last one that we're going to talk about is chemotrophs. So these guys actually get their energy from a redox reaction, so an oxidation reduction reaction. And an oxidation reduction reaction, it happens whenever electrons are removed from a compound, and then they utilize that energy for themselves. So a chemical reaction has to occur in order for a chemotroph to survive. And there are two main types. There are lithotrophs and organotrophs. So I'm going to move to the next slide to show you. So this is kind of like a breakdown of all of them. So chemotrophs, they kind of get separated into lithotrophs and organotrophs, like I said. So with chemotrophs, they can either they can do their chemical reaction by using inorganic molecules or organic molecules. So from this, this is just a summary of all of the trophies that we talked about on the previous slide. So biggest thing I want you to get from this is literally just what's the difference between the six. Okay? Okie dokie. So now we're going to transition into the next concept, which is the culture and counting of bacteria. So how do we grow and culture separate bacteria in the laboratory? So we did that actually today in lab on Tuesday. So ideally what you want is to get a pure culture. So this is a single genetically homogeneous strand of a specific species. So that's whenever I was talking about like how you want a single colony. So that is considered a pure culture because ideally it arose from one single cell and it only came from that specific species. You can grow them in liquid or you can grow them on solid media, which is what we did today. So we played with both. And then after you get a pure culture, you examine it, and then also you can use specific medias to actually separate out different organisms, as you see in this photo. So this photo has multiple species on it, but based on how they grew, you can distinguish them. And we'll actually talk more about that in lab whenever we get to differentiating between gram-positive and gram-negative bacteria. So the most versatile media that you can use is called agar. So that is, it's like Jello, and it is it's literally like a little meal for your bacteria babies. And we talked about different ways of getting it. So there's the isolation streaking, which is actually what we did today. And then there's plate spreading, which is where you have a diluted culture. So you have some bacteria, but it's diluted with a lot of media. And then what you do is put a little drop on your plate, and then you just let the liquid spread around. Or what you can do is do something that's called a pour plate, which is where you literally just pour your culture onto a plate and you just let it grow. But the best way, though, to get a pure colony is isolation streaking or quadrant streaking. That's the best thing to do. Okay. So there are different media types, and we'll actually go a little bit more into detail in lab, but I do want you to know the big picture of these media. So there's general purpose, which is the one that we used in lab today, which is the nutrient agar. So that is the one that can grow any microbe, and you primarily just want to isolate it. So you just want a pure colony from that. There's the enriched media, which has growth factors added, so such as something like for a fact that a specific bacteria likes to grow in the blood. You can add blood into the agar in order to promote growth for a particular bacteria. So that's a little bit more narrow in media type, whereas general purpose, it'll pretty much grow anything. And then you have selective and enriched. So this is designed to inhibit something that you don't want to be growing on it. So an example of that can be like a plate that is designed for only growing gram-negative bacteria. And we'll go more into detail of that in lab, but just know in the context for lecture, just know the differences between them, okay? And then also, the last one is differential or indicator. So this is the one that is designed to differentiate between different bacterias. It allows growth of more than one organism, but a lot of times they are morphologically different, meaning that they look different on the plate, such as the image before. So as you see right here, so this might have been a differential indicator media, because as you see, like, we have different growth types. So we have two bacterial colonies right here, but they look very different. Whereas one is more orangey yellow and very smooth looking, whereas this one is very wrinkled in appearance and yellow. So that's another way of telling for differential media. And then also just know that the parameters of media is not, it's not very rigid. A certain media type can technically be both, okay? So just know that it's not a one size fits all, okay? So they can bleed over into different medias. So one media might specifically be selective and differential. So an example of a media that is selective and differential is called McCorney or just MAC. So we'll talk more about MAC in lab. Just know that it's not very rigid. So medias can bleed over into other categories, okay? Okie dokie. So next, we're going to talk about how do we actually count these cells once they're grown. So once we have these bad boys, what do we do with them? So there are multiple ways of doing it. For us, we're actually not going to do this in lab, but just know that there are different ways of doing it. So you can count living and dead cells at the same time. It's something called a hemocytometer. So this is a specific microscope slide that actually has little chambers on it. As you see, like, has little ridges in it. And what you do is that you put this underneath a microscope under some kind of magnification, and then you literally just hand count them. So what you do, like, you'll see, like, on the slide, it creates this little box. And what you do is that you count the area within that box, and then you average the surrounding boxes, and that'll give you a rough estimate of how much bacteria you have in your culture. Or there's a direct way where you get a very specific number, and that's without a microscope. So this is done with a fax machine, and this counts and separates bacteria cells based on different properties. So just know that there's different ways of counting cells. There's direct counting where you physically hand count it, and then there's another way of doing it without a microscope, and that's using the fax machine. So this is variable counting. So to obtain a variable count, so like a guesstimation, what you do is that you dilute liquid cultures. So what you'll do is that you'll have a very, very concentrate, and what you do is that you do something called a serial dilution. So what you'll have is that you'll have your original media that you had, your original stock solution, and then you'll have other media that haven't been inoculated yet. So what you'll do is that you'll take a ml from this one and then put it into your next broth, grow it up for a little bit, and then what you'll do, you'll let it incubate maybe for like 30 minutes or so, and then you'll take a ml from this one, put it again into another one, and then continue that process So you get a very dilute culture where there's minimal bacteria, so it's easier to obtain a pure colony, whereas from here, it is a complete um, garden of bacteria. So it's just one big lawn of bacteria, whereas you want something a little bit closer to the last two, where it's easier to get a pure colony. So we talked about direct and variable counting. Now we're going to talk about indirect methods. So we will do this in lab actually. So this one we will do is optical density. So this is how dense is your culture. So how much light can go through your culture. So theoretically speaking, if light can travel through your culture, you don't have a lot. Whereas if light gets bounced off and not absorbed as much, then you have a very dense culture. So meaning that you have a lot of bacterial growth, as you see in this image. So all the light goes through, you can see that there's not a lot of bacteria, whereas in this one, only a little light goes through and there's a lot of bacteria. So you'll see that, and you read it as absorbency. So if you have more absorbency, then you have less bacterial growth. And this is called turbidity. So turbidity has to do with how much light can pass through the sample. Okay, so that's a method of indirect counting. So it's kind of just like a scale. It's like 10 being the most thick and 1 being the least thick amount of culture. So it's not a true number, but it gives you an idea. Okay, so now we're going to transition into the last little bit about the bacterial growth cycle. So what determines the rate of microbial growth? So there's a lot of things that can determine it, but the ultimate goal of any species is to make more of itself, right? So we all want little me's running around. And the way bacteria does that is that they do something called binary fusion. So this is where they make a copy of themselves and then they split into equal cells. So remember, bacteria do not have sex. They don't have sex, okay? I can't say that enough. Please do not say bacteria have sex. They undergo binary fusion where they have an original cell, and then that original cell will start duplicating itself, it'll start cloning itself, and then it will eventually split off at this division point, as you see right here. So it will eventually cleave off, and then you'll have two copies. So there are times where binary fusion can be symmetrical, which is more like the first picture, or it could be a little bit more asymmetrical where you have like a budding cell like this. So either way, it's still binary fusion. They still retain the same amount of genomic information, just different sizes. So eukaryotic microbes, such as like fungi, they actually undergo true mitosis, but they do not do sexual reproduction. They can, but not a lot of them. There's only a handful, but we're just going to talk about the ones that don't do sexual reproduction. So on the test, they don't undergo sexual reproduction, okay guys? It's all asexual and binary fission, excuse me. Okay, so this is the general growth cycle of any pathogen. So for the test, I definitely want you to know this. So there are 4 main phases of the growth that a microbe will undergo. There's the lag, the log, stationary, and death. So I do want you to know the differences between each one. So lag is the first one. So this is where the bacteria is preparing to make more of itself. And log is where there's exponential growth of them, like where they're going like crazy. It's normally almost a straight line. It's just exponential growth of them. So they're going like crazy. And then at some point, the cell will get a signal to stop growing. And what will happen is that it will tell itself, stop growing. They're starting to stress out because there's too many of them in a specific area. So this is called the stationary phase, where the growth kind of like tapers off. And then eventually what will happen is the death phase. So this is where the cells begin to die, and it does kind of go at an exponential rate as well, but not as steep as the log phase. So once they hit the stationary phase, what will happen in order for them to enter the death phase is that there's no more nutrition for them. So they just start dying off, they start starving, they're starting to compete with each other. So since they're in a competition phase, they just want to survive, but unfortunately they can't because there's not enough nutrients for them, so they start dying off. So definitely know the four phases and know what each phase represents. Okay? Okay, so now we're going to talk about the environment, how that affects and limits microbial growth. So like I said before, there are a lot of factors that can play into limiting microbial growth. So right now we're going to talk about the specific environment, such as temperature. So um, environmental factors that affect microbes, it's very dependent on the species. Some microbes, they only survive well in acidic environments, and other microbes, they survive better in a basic environment. It just depends on the species. A key term for y'all to know is extremophils. So these are the bacteria, archaea, and a handful of eukaryotes, not a lot, that live in the extreme environments, such as high temperature or low temperature, the extreme pHs, so extremely acidic, extremely basic, or places of high pressure, so those are specifically called barophils, or with osmolarity, so these are halophils. So these are the ones that like salty concentration, so NaCl is salt. And then also every protein and molecular structure within the cell is affected by its environment. So just know that everything plays a role. Everything's intertwined, okay, guys? So we're going to go a little bit more into detail about the temperature ones. So these are classifications of organisms where they grow within certain temperature ranges. So there's cytrophils if they grow within this temperature range. So they like to grow in a more cooler environment. And then there's mesophils. They like to grow in the more warmer environment. So the 10 to 45 degrees centigrade. Then there's the thermophils, which like higher temperature. So that's the 40 to 7 range centigrade. And then there's the hyperthermophils, which is where they can grow just in extreme heats. So most bacteria is that we're going to be dealing with are the mesophils. And the reason why most microbes are mesophils is because our body is actually 37 degrees centigrade. So that's roughly 98.5-ish Fahrenheit. So that is where they like to grow. They love to grow at around 37. They can grow at room temperature, so that's room temperature is approximately like 25 degrees centigrade. So most bacteria, most microbes in general are mesophils, solely based on the fact that majority of microbes that infect humans, they like to be mesophils because humans are in the mesophil range. So our bodies are 37. 37 falls within this range. So majority of microbes that infect humans are considered mesophils. So now we're going to talk about bacteria that can live with or without oxygen. So aerobic means that they like to use oxygen. They prefer the presence of oxygen, whereas anaerobic, this is where they prefer no oxygen in their environment. Oh, excuse me. So anaerobic, no oxygen. Aerobic prefers oxygen in order to in their environment, excuse me. So they prefer oxygen or not prefer oxygen in their environment. Okay. So the Earth's atmosphere it has a good amount of oxygen, and we rely to breathe on it, but do all bacteria like to grow in the presence of oxygen? Anaerobes, yes, but aerobics, no. So what happens? Aerobes, where they prefer oxygen, they destroy something that's called ROS, and with that, it aids in enzymes. So they'll use specific enzymes such as the superoxidase or peroxidase or catalase. So if if aerobic bacteria do not have oxygen, they are unable to utilize their enzymes, and they're unable to remove hydrogen peroxide, and they're unable to remove superoxides from their environment. So with that, they're unable to survive because they can't metabolize anything because of the fact that oxygen's not present for them, and they need oxygen to metabolize. So that's the big thing, guys. And then there are different types of anaerobes. I'm actually not going to cover this in lecture because we're going to cover this more in detail in lab. So for the exam, you don't need to know this, but definitely know this part of where aerobic microbes, where they need oxygen so they can destroy ROS with the aid of using enzymes. So definitely know that concept, but you don't need to know this figure for lecture. But for lab, it will come up again. So definitely for lab, you need to know this later on, okay? Okie dokie. So how do we culture anaerobic bacteria if they don't like oxygen? So we are not going to do the techniques that are on this slide, but it is good for you to know because we will be creating an anaerobic environment later on in term. So we are going to use something kind of similar to this gas jar, but instead of a jar, it's just going to be a pouch. So with the jar or with the pouch, what they do is that they remove oxygen, and then so CO2 can be generated for these microbes so they can grow. So there are mainly one bacteria that we're going to deal with that does prefer a anaerobic environment. That's Clostridium. He's the main one that prefers the anaerobic environment. But like I said, we're not going to use these jars. So these jars, like you actually like pump, you kind of pump them so you suck out all the oxygen. Or there's also these chambers, but this is mainly in research chambers where you have this airlock that makes sure that air doesn't get in, and then it has a vacuum, so it sucks it out as well. So just know that this is how you create an anaerobic environment, but specifically in lab, we're going to be using something a little bit closer to the jar where we inoculate our plates like how we did today in class, and then you put them inside essentially like a sandwich bag. You push out all the air, you pump out all the air and everything, and then you put in this tablet. So you put in this tablet in order for an indicator so you know that you created a true anaerobic environment. And a lot of times what will happen is if it wasn't an anaerobic environment, the tablet will change color. I believe our tablets are blue, but sometimes they can also be pink. But just know that there's a color change within the tablet, and if there's a color change in that indicator tablet for the pouch, then a true anaerobic environment was not created. Okay, so now we're going to move into microbial communities and differentiation between cells. So do bacteria cooperate with each other? Absolutely. Bacteria talk to each other, guys. Do we actually know how they talk to each other? A little bit. We know some mechanisms, but not all. And the biggest way of bacterial cooperation is by making a biofilm. So we dabbled a little bit in this, but a biofilm is a mass of cells that stick and multiply to a surface. So an example of that is like plaque. So if you have excessive plaque, that is considered a biofilm, as well as the stickiness within a cystic fibrosis patient. So that is also considered a biofilm. Also, with biofilms, they don't have to be just one species. They can be multiple species. So, but a lot of times an initial biofilm will happen with one, and then another bacteria or even a fungus can will come in and they will cohabitate with each other a lot of times, which is pretty cool in my opinion. Um, but in order for a biofilm to happen, there has to be surface attachment. Okay? If these bacteria cannot attach or adhere to a surface, then the likelihood of a biofilm forming is very minimal. Okay? So here is an example of a biofilm of like how it starts. So it kind of goes through like this little process where we have cells and everything. They create like this little layer, this initial layer on a surface of any kind. It can be like on a tissue or even in a plate. And then what will happen is that it'll start forming and multiplying and everything. And then these cells will start sticking to each other. They'll start producing a lot of polysaccharides, and then it fully matures whenever it becomes very thick and rich. It doesn't have to just be one species, it can be multiples. And then what will happen is that once the biofilm completely has matured, a lot of times it'll somewhat dissolve a little bit, and then it will disperse some organisms for them to continue to spread. Or whenever the biofilm is dissolving, that could also mean the presence of a medication. But biofilms, they are a virulence factor because if you think about it, it's very easy to penetrate individual little cells. But if you have to penetrate through this whole thing, it's a lot harder to penetrate, okay? So remember, biofilms is a virulence factor, so it makes, if a biofilm is formed, it makes it harder to treat because it's like a extra layer of protection. And then also, the way biofilms are formed is through quorum sensing. So in biology, quorum sensing is the ability for a cell to detect and respond to cellular population based on density. So it sees that it's not that dense right here, so it's just like, oh, I'll make more of myself. I'll make more and more and more. And then eventually it'll reach a certain density where it's just like, okay, we need to pop off and go somewhere else because we're too dense right here. So it wants to spread out, and they do that through quorum sensing. And they also do gene regulation too. And then whenever a bacteria has gathered a sufficient amount of numbers, so like enough population, they'll start acting like a multicellular organism rather than being single cellular, which is how they normally are. So pretty cool, guys. So there's another thing called endospores, excuse me, in suspended animation. So these are kind of like, they're in dormant, like they're kind of hibernating. So there are certain bacteria that have the ability to pretty much go like hibernate whenever they are in extreme heat, or whenever they're in like not a so desirable environment. So mainly it's heat. These spores are very resistant to heat. An example that can do that is C. botulism, and then also B. cereus. Like we actually played with him a little bit today in class. So B. cereus does create spores. And a lot of times, if they can produce these endosperms, they are a very, very hardy bacteria. So just a little side note right there, guys, where sometimes bacteria can go dormant, and whenever they are in a dormant or like suspended state, they're called spores. And the reason why they produce these spores is because they're in a very intolerant environment, and primarily that's a very heated environment. Okay, guys, so that will wrap up our chapter 6 lecture. So we covered nutrition, how nutrition in the environment can impact microbial growth, and then also how they differentiate. We talked about nutrition and the environmental limits, such as like mesophils, extremophils, and everything. We talked about various media types. We talked about four main types, and also that media types, they're not very rigid. Some medias can bleed into multiple types. We talked about the bacterial growth cycle, so the log, lag, exponential, and stationary death phase. So we talked about that. So definitely know that for your test, guys. And we talked about how important understanding microbial growth can be, and then also biofilms and why biofilms are important for infectious diseases, such as they are a very strong virulence factor, and why they are a virulence factor because they're harder to get medicine into because they're very thick and very dense. Okay, guys, I hope you enjoyed this lecture. I'll see y'all soon.